Hi, I'm Abrar Hussain from University of Hyderabad, India, from NEF Lab. So I am going to present some talk about characterization of photocytes derived from proteins, and my emphasis will be on CD2AP. So before starting, we know our kidneys are very important, playing role in the filtration of blood and formation of urine. And in the kidneys, there are nephrons, which are structural and functional units. And in the nephron, the actual filtration process lies in the glomerular part of the kidney. So this glomerular part of the kidney, when we are deeping diver, we are seeing there are three-layer filtration process taking place. One is the endothelial layer, which is the innermost. Then GBM is there, glomerular basement membrane. Then there are terminally differentiated epithelial cells, which are terminally differentiated. And during the developmental stage, these cells lead to the formation of primary food processes. These primary food processes then further divide and they lead to the formation of secondary ones. And secondary ones are one podocyte and other interlace, and they lead to the formation of a junction that's called ciliate diaphragm. This is the TEM image. Here we can show how three layers are in concert, playing very important role in the filtration of blood and formation of urine, which is devoid of proteins. So this is the actual macromolecular complex or ciliate diaphragm, which is attaining the dimensions of 30 to 40 nanometer. And these dimensions of pore size won't allow the movement of albumin from blood to go into urine. So when we are deeply driving here, we are seeing that there are 5 to 6 proteins playing role in the formation of complex. Some are membrane proteins like this nephrin, Nef1, TRPC6, podosin, and this CD2AP protein is actually an adapter protein. From one side, it is interacting with the cytosolic portions of membrane protein, which are forming complex, and from other side, it is interacting with the actin cytoskeleton of cells. So this is very crucial protein for the entire complex. So any changes in these proteins, whether mutation, can lead to the dissociation of one complex, and it will facilitate the migration of proteins from blood into the urine. So here we can see how the three layers are how the three layers are intact, and there is no movement of proteins like albumin from blood to go into urine. But in disease condition, we can see the outermost layer, that means ciliate diaphragm, is lost. So when there is no ciliate diaphragm, the movement of albumin will go from blood, it will go into urine, and it will lead to the development of nephrotic syndrome, which is characterized by proteinuria and edema. Further, there is no effective treatment. To some extent, cortisols are being given, but after some time, there's lapse of disease. So till date, we know that this nephrin, podosin, CD2AP, TRPC6, they co-localize at ST, and the co-immunoprecipitation studies also reveal that these interact and they form complex. And there are physiological and cellular studies which reveal that mutation in these proteins can lead to the proteinuria, disease development, and some mutation do affect the co-localization of proteins. But the key questions are there are no information available about interface residues, which are responsible for complex formation. Do mutations influence the protein structure or complex? There is no clue about it. Structural details of SD proteins, whether individual or complex, is yet to be elucidated. The reason for this may be because these proteins are not normal kits like the globular proteins. They are intrinsically disordered proteins, but they possess high functional pertinence. They can help in signal transduction, interactions. They can mediate complex formation. Additionally, they can bind to different partners, enable the distinct conformations, which in turn enable it to interact with different targets. So here I characterized, expressed, purified, and characterized this CD-TAP wild type protein. So we used PET28A overexpression vector in which our gene was cloned between NDL1 and Jo1 sites, followed by induction conformation by different concentrations of IPTG with 0.2, 0.5, 0.7, and 1. Here, finally, we concluded that 1 millimolar IPTG is showing good induction, and there is also good solubility of the protein of interest. Then finally, after solubilization, we purified the protein by affinity chromatography. Here we can see marker followed by pre-induction, post-induction. This is solubilization of the protein. Then this is Watts fractions, and finally the protein of interest was purified to near homogeneity corresponding to band 73 kD. After successful purification, we were interested to find out the oligomeric nature of the protein because there were speculations about the existence of SD proteins in oligomeric forms, but till date there is no information. So we were interested and we did size exclusion chromatography. Before that, we generated the standard plot of the molecular markers. Then finally we injected the protein of interest, and from this chromatogram we are seeing that the protein of interest is forming higher order oligomer. We don't know this because we were not having marker. but there is one more peak that is corresponding to a 9 mer. So thus the corresponding fractions were further run on the gel, which tabulate the corresponding molecular weight. Thus we can summarize that this CDTAP in isolation is adapting 9 mer conformation. Next, we investigated the secondary structural content of the protein by circular dichroism spectroscopy. So before discussing this unique spectrum we first tried to see into the AlphaFold database. There we are finding majority of the protein in monomeric is intrinsically disordered, followed by small percentage of alpha helix and beta sheets. So when we are comparing with our unique spectrum, we are seeing the parallels what we are getting in AlphaFold. There are majority of the protein is IDR, followed by small percentage of helix and sheets. Next, we investigated the existence of polypeptide chain in tertiary structure. So we are seeing that we are getting broad signals 260 to 295, 300 nanometer, which is most probably contributed by tyrosine, phenylalanine, and tryptophan together. And also fluorescence emission give a very strong signal at 339 nanometer, which further speculate the existence of protein in tertiary structure. So next, we were interested to see how the temperature is influencing the protein oligomers as well as quaternary tertiary structure of the protein. So for that, we did intrinsic tryptophan fluorescence. So here we are seeing in this at 5 degree we are getting maximum emission. Then with increased temperature, the emission intensity is decreasing. When we try to plot the maximum emission with increased temperature, we are seeing linear transition going with the temperature. Most probably the oligomers are dissociating and the hydrophobic cores gets exposed to environment. And it is not the typical sigmoidal transition commonly observed in globular proteins. Further, it was interesting to see when we reversed the temperature conditions, we found that there was refolding nature of the protein, but we can't anticipate that with the native refolding of the protein. Further, we investigated the thermal stability of secondary structural content, whether IDRs, alpha helix, or beta sheet with thermal melt assays. We are seeing that with increasing temperature, there is refolding of alpha helix taking place to some extent, but after 65, there is quite thermal stability of the secondary structures. And the IDRs, intrinsically disordered regions, are losing the structure to some extent up to 65, beyond that all the structures are robust at high temperature. Thus, we can summarize that this protein was effectively expressed by 1 millimolar IPDG concentration. The purification protocol was first time optimized and the protein was purified in near homogeneity. Analytical experiments revealed the oligomeric nature of protein pertaining to 9-mer. And secondary structural elements, IDRs, are dominating the secondary structural content, followed by alpha helix and sheet. And this protein has modest quaternary and tertiary structure, and secondary structural elements were robust at higher temperatures. I recently published this story, and I acknowledge University of Hyderabad, our NEF lab, and finally P12 meeting. Thank you for listening.